In what many once believed to be an unreachable frontier of neuroscience, a remarkable team of scientists has achieved a feat so intricate and ambitious that it not only defies expectations, it opens a stunning new chapter in our understanding of the brain. Using a sliver of mouse brain tissue no larger than a grain of sand, researchers have crafted the most detailed three-dimensional map of a mammalian brain ever produced, capturing a level of precision, complexity, and beauty that was previously thought to be beyond the limits of science and technology. The breakthrough represents a decade-long collaboration among 150 scientists from 22 institutions, led by the Allen Institute for Brain Science, Baylor College of Medicine, and Princeton University. This project, known as Microns, Machine Intelligence from Cortical Networks, resulted in a dataset so immense, 1.6 petabytes, that it rivals 22 years of non-stop HD video. Yet this enormous archive emerged from just 1 cubic millimeter of mouse visual cortex, a region responsible for processing what the animal sees. Within this single speck of tissue, scientists traced 84,000 neurons, 200,000 brain cells, over 500 million synapses, and more than 3 miles of neuronal wiring. The sheer scale is breathtaking, and the fact that all of this complexity resides in such a minuscule volume is a testament to the incredible density and elegance of the brain's architecture. What makes this achievement even more inspiring is that it proves wrong a notion held by some of the greatest minds in science. In 1979, Nobel laureate Francis Crick famously wrote that asking for an exact wiring diagram of a cubic millimeter of brain tissue was akin to asking for the impossible. And yet, here we are in 2025, with a beautifully detailed, colorized map showing not only how neurons are shaped and arranged, but also how they interact, communicate, and fire in real time as a living creature experiences the world. The analogy made by Dr. Forrest Kallman of the Allen Institute, that studying this map is akin to gazing at a distant galaxy, feels especially apparent. It's a fusion of science and wonder, where data meets artistry, and where complexity reveals elegance rather than confusion. The process behind this achievement was an extraordinary combination of cutting-edge biology, machine learning, and collaborative engineering. First, researchers recorded brain activity while the mouse was awake and visually stimulated by a curated playlist of high-action movie scenes, from The Matrix to extreme sports videos. After imaging how neurons fired in response, the mouse brain was preserved and sliced into over 28,000 ultra-thin sections. Each slice was then photographed and digitally stitched together, with advanced artificial intelligence algorithms tracing the intricate threads of each neuron through the layers. These AI-generated maps were carefully reviewed and refined by human experts, ensuring accuracy while pushing the boundaries of digital neuroscience. What makes this more than just a technical achievement is the promise it holds for the future. This mouse brain connectome, a full-circuit map of how neurons link and communicate, has been made publicly accessible, empowering scientists around the world to explore, analyze, and build upon the data. It's not just a snapshot of a single brain, it's a powerful new tool for decoding the very foundations of cognition, perception, learning, and memory. And while it may be only a fragment of the full mouse brain, the roadmap to scaling up is now clearer than ever. Researchers believe that mapping the entire connectome of a mouse is now within reach, perhaps achievable in just a few years. What once seemed unthinkable is quickly becoming a matter of when, not if. The implications stretch far beyond rodents. Understanding how neurons connect and misconnect has direct relevance for studying brain disorders such as Alzheimer's, autism, schizophrenia, and Parkinson's disease. These are illnesses rooted in the disorganization or disruption of neural circuits, and now scientists have the equivalent of a Google map for exploring and comparing healthy and diseased brain tissue. As Dr. Nuno Macarico da Costa put it, having a circuit diagram for a malfunctioning radio makes it far easier to repair. The same logic applies to the brain. And since lab mice are already standard models for studying human diseases, the refined understanding of their neural architecture could translate into more precise, effective treatments for human conditions. Even more heartening is the notion that this map doesn't merely represent knowledge, it represents a shift in what's possible. The creation of a digitized, searchable, Interactable Brain Blueprint shows what can happen when technology and curiosity intersect. With just a few keystrokes, a researcher can now access data that might once have required years of painstaking manual study. This democratization of discovery lowers barriers, accelerates collaboration, and allows imagination to flourish in ways that once belonged only to science fiction. Of course, mapping a human brain in the same way remains a monumental challenge. The human brain is 1,500 times larger than that of a mouse and introduces vast ethical, technical, and computational hurdles. Yet, even here, there is a quiet confidence among the research community. Axonal mapping, tracing the longer, signal-transmitting arms of neurons, may be feasible in the nearer term, and synaptic resolution mapping might one day follow. In every respect, this mouse-brain connectome is not an endpoint but a beginning, a model that can be refined, expanded, and eventually applied to even more complex systems. Ultimately, this achievement is a celebration of the possible. It reveals the astonishing intricacy of nature's most powerful organ, while showing that with patience, creativity, and collaboration, even the most ambitious dreams in science can be realized. It suggests that the mysteries of the mind are not locked behind an impenetrable wall. They are waiting to be unraveled, one neuron at a time. And in doing so, we're not just mapping the brain, we're redefining the limits of what humanity can understand and achieve.